I saw focused ultrasound as a unique tool to help unlock barriers that had been sort of unsurmountable for many, many years related to treating these tumors effectively.
Welcome to Curing with Sound, a podcast presented by the Focus Ultrasound Foundation. I'm your host, Allison Preston Smith. Focus Ultrasound is showing immense promise in the treatment of brain tumors. One of the biggest challenges in treating brain disease is the blood-brain barrier. Today's guest, Dr. Graeme Woodworth, is a neurosurgeon, researcher, and chair of the Department of Neurosurgery at the University of Maryland School of Medicine. He has been at the forefront of efforts to use focus ultrasound and microbubbles to safely and temporarily open the blood-brain barrier, making it possible to deliver therapies directly to the brain and benefiting patients with a variety of conditions. Last August, he and his team published findings on a reliable way to measure and predict blood-brain barrier opening using acoustic emissions. More recently, Dr. Woodworth and his co-investigators published results from a glioblastoma clinical trial in Lancet Oncology, where focus ultrasound was used to open the blood-brain barrier, allowing for enhanced delivery of chemotherapy to the brain. Welcome to the podcast, Graeme.
Allison, thank you so much for having me.
What first motivated you to work in focus ultrasound, and more specifically on blood-brain barrier opening?
Well, my interest in focused ultrasound really came to be from my interest in improving therapies and treatments for patients with brain tumors. And I saw focused ultrasound as a unique tool to help unlock barriers that had been sort of unsurmountable for many, many years related to treating these tumors effectively. And it was in that research that we were doing back many years ago now, where focused ultrasound was just sort of coming back into the world of neurosciences, because it had really been around many years before in the 1950s and 60s when it was being studied for neuromodulation, and then sort of left the field as other therapies, other directed energy therapies took over. But focused ultrasound came back into the sort of scientific realm, not quite the clinical realm yet, but in the scientific realm. And we started to study it for improving drug delivery for brain tumors, and that became very exciting.
What role do microbubbles play in blood-brain barrier opening?
So with focused ultrasound, it can be used alone in various modes. It can be used in modes that are more mechanical, and it can be used in modes that are more thermal or heat generating. And that's one of the things that makes ultrasound such an exciting energy form, is it can be tuned across these different modes to achieve different purposes and different end biological effects, and then also relate those biological effects to specific therapeutic effects. When it comes to using ultrasound with microbubbles, the idea is that the bubbles themselves become resonators in the acoustic field, and that resonating forces that are created by the bubbles in the acoustic field as they expand and contract and oscillate actually create mechanical effects on the blood vessels. The bubbles are within the vasculature of the body, and when the energy is directed across the brain, those bubbles are resonating within the brain blood vessels, and it's that stable resonance of the bubbles in the brain blood vessels that leads to temporary blood-brain barrier opening.
In your recent study published in Device, you and your team looked at data from glioblastoma patients to establish a method for measuring and predicting blood-brain barrier opening with focus ultrasound. Can you briefly summarize that study and the importance of your findings?
Yes. So the idea with ultrasound in many ways is that by applying the energy, you're achieving an end bioeffect in the region of interest, in this case, the brain. But we want to be able to measure that effect in real time so we know how much energy has been delivered to the region and what effects have been generated there. We don't want to just be aiming the energy into the brain and not knowing how much has gotten there or how much of this bubble oscillation resonance has occurred. So taking a step back to other forms of ultrasound that I mentioned earlier, this idea of using ultrasound in thermal modes has been used for years now to treat dysfunctional neural circuits in the brain for conditions like essential tremor. And the use of ultrasound in that mode has really been propelled by the ability to measure in real time thermal change, the temperature change that happens in the targeted region. And that creates an ability to know in real time that the energy has been delivered to a specific target at a given amount, or you could even say dose. So with the bubble form of ultrasound, microbubble enhanced focus ultrasound, what we were looking for is a way to achieve a similar real-time measurement of the end effect. In this case, that end effect is not temperature change. It is the bubble oscillation, the stable bubble oscillation in the blood vessels. So it turns out when the bubbles are oscillating in the blood vessels, they send off an echo signal back. And that echo signal can be detected in real time by what are called passive cavitation detectors, which forms an acoustic emissions monitoring system. So that system can be used to monitor both the safety of the ultrasound treatment, but also monitor this bubble oscillation, which can be converted in a sense into a dose. And it's that closed feedback loop system of energy being delivered in, the emissions coming out and being measured in real time that allow you to create the desired bioeffect, in this case, the neurovasomodulation that leads to blood-brain barrier opening. We correlated that measured effect, that measured dose, with the blood-brain barrier opening in this new paper, and found an interesting correlation that was nonlinear. It was actually a correlation that uncovered a dynamic range wherein the dose, where that range occurs, actually is where you have the greatest opening of the blood-brain barrier and change in the vasculature. Outside of that range, there's really no additional blood-brain barrier opening and really no initiation of blood-brain barrier opening before it.
And before this discovery, how did doctors know whether the blood-brain barrier had been successfully opened, and what problem does your new method solve?
So there are many ways to measure whether the blood-brain barrier has been opened, such as giving an imaging contrast agent, one that's used in the hospital and the clinic and in scientific areas all the time is gadolinium enhanced MRI scans. So doing an imaging study after the focused ultrasound treatment can be a method for assessing whether the blood-brain barrier has been opened, because we see a dye or an imaging contrast agent be moving from the blood vessel into the tissues around it. So that's one method. A similar method would be using an intravascular dye that would otherwise not move out of the blood vessel into the tissues unless the blood vessel has been temporarily permeabilized or disrupted. Those dyes that have been used are both fluorescent and visible dyes. So these are ways that can be measured after focused ultrasound has been applied. But to be able to measure in real time that you're generating the acoustic effects and the bubble oscillation that lead to blood-brain barrier opening, and not having to necessarily inject a dye or measure something in the tissues after the effect, is the big difference here. It's a real-time monitoring system and dosing strategy that can be used to then predict that the blood-brain barrier has been opened and not do it afterwards.
Why does having a more accurate way to measure blood-brain barrier opening matter for patients right now?
Well, one of the things that's going to be changing, I believe, in the next few years is there's going to be many more devices that are going to be coming into the clinical arena, not just the research arena, but into the clinical arena for performing blood-brain barrier opening using a similar technique with the bubbles and the ultrasound energy. The question really is going to be for us as we apply these new devices in patients is, how do we standardize these treatments? How do we relate a given focused ultrasound treatment with one device to another focused ultrasound treatment with a different device? If in the end we want to get to the same point of achieving blood-brain barrier opening in a reproducible, rigorous way that's measurable in real time and relatable, whether it's with one device or another, we need an end measure and dosing strategy to achieve that kind of standardization and clinical and scientific rigor. That's going to be critical as we begin applying technologies like this for drug delivery or other therapeutic effects in the brain. because we can't just say we've opened the blood-brain barrier as measured by gadolinium and say that's the same as, another device or another technique. We need a standardized way of relating the treatments and dosing the treatments so that they can be repeatable, consistent, and comparable.
You've also recently published the results of a multicenter glioblastoma clinical trial in Lancet Oncology. This study used focused ultrasound to open the blood-brain barrier for enhanced drug delivery of chemotherapy. So can you tell us about this study and your findings?
Yeah, so this study was a very exciting study on many levels. At the first level, it was exciting that the FDA allowed us to use focused ultrasound for the first time in patients receiving the standard chemotherapy that's used to treat patients with high-grade gliomas. This is the chemotherapy called temozolomide, which is an orally administered chemotherapy. And the FDA allowed us to use the focused ultrasound during the standard chemotherapy cycles, so monthly treatments for 6 months, which was the first time, that we were going to be able to use the technology in a repeated way in the same patient during chemotherapy. So the initial focus of this study, given it was the first time this was being done, was the safety and feasibility of doing that, which was quite a challenge, to be able to take patients through this each month and get the device and the systems and the protocols organized in such a way that it was repeatable, safe, that the patients weren't in a tremendous amount of discomfort in any way. We didn't want it to take long periods of time. Of course, we wanted to be careful, but all of these things had to be worked out. So the study was very exciting on all of those levels. What we didn't know is whether we would see any improvement in survival or improvement in drug delivery. But what we were able to do was compare the outcomes of these patients, both from an imaging perspective and an overall survival perspective, to a matched control group that was done independent of the study team by a biostatistics team at Dana-Farber Cancer Institute and Brigham and Women's Hospital. And with this matched control group, we were able to assess whether the patients in the study across the 5 sites had improvement in tumor control as measured by MRI and or improvement in overall survival. And in both instances, we found a significant improvement in both tumor control and overall survival, suggesting that adding focused ultrasound to standard chemotherapy cycles in patients with newly diagnosed high-grade gliomas improves their outcomes. And that's a very exciting signal that this technology may really benefit patients, especially when we start to think about chemotherapies that aren't otherwise used for brain tumors like this, because they're not known to get into the brain in effective concentrations without causing significant systemic side effects. So this really begins to open the door for us to think differently about the types of drugs, the types of therapies that we can apply for these patients earlier in the disease management, before they recur, because the study was done before tumor recurrence, after maximal safe surgery and standard of care radiochemotherapy. That's when these focused ultrasound plus drug treatments occurred.
So will there be another clinical trial expanding on the findings?
So there are numerous other clinical trials that we're thinking about with the device, with the focused ultrasound device. There are other devices out there in the world as well that are being combined with drugs of various types. For us, we are looking to specifically partner with pharma companies, biotech companies that have exciting drugs that may have been tested in other solid tumors and in some cases brain tumors, but may not have worked well because they didn't get into the brain in high enough concentrations. And we believe that we have a unique solution to pair and partner with those drugs and these companies to create effective drug-device combinations.
You mentioned the importance of finding the most effective drug-device combinations as efficiently as possible. What kind of strategies are being used to speed up that process?
Yes, I think that's a great question, because we don't want this to take 10 years. We want this to take as little time as possible to uncover what could be the most effective drug device combinations. So some of the unique ways we're thinking about that is, first, are there companion diagnostics that we can use with the drug device combinations that would allow us to see a signal of whether the drug device combination is working without having to wait for the tumor to recur by using MRI to gauge that, or waiting to see if the patient survives longer to gauge that. So are there companion diagnostics that can be used along the way? An example of that would be a T-cell imaging agent. So if we're going to be using focused ultrasound to improve the delivery of an immunotherapeutic that would otherwise, based on preclinical testing and other mechanistic understanding of the therapy, lead to an infiltration of anti-tumor T cells, such as CD8 T cells. If we could measure that infiltration of CD8 T cells after the drug device combination therapy, that would give us an earlier signal that it's working. That's one example. Another example would be using the drug device combination therapy before surgery in so-called window of opportunity studies, where we would use or deliver the therapy, say, 1 to 2 weeks before a planned surgery, to then use the surgery and the analysis of the resected tumor tissues that have been treated with the drug device combination to assess the early responses and biological effects that that combination therapy was having in that tumor or in the tumor region. So these are ideas that we are actively exploring to try to accelerate the development and the assessment of the biology of therapeutic responses without having to wait years for survival or imaging signals that are traditionally used in these studies.
And you alluded to this earlier, but how close are we to seeing this technology as part of routine care for patients, and what steps still need to happen to get there?
Well, the first step would be to get to a point where one of these devices is approved for clinical use by the FDA and has a reimbursable code where it could be used. So our strategy so far related to that with this device has been not a therapeutic strategy, but a diagnostic strategy. And the reason for that is having a diagnostic strategy focuses on the device as opposed to a therapeutic strategy, which is more complicated, where the device plus drug is being approved. So it turns out that having the blood-brain barrier opened can also release biomarkers from the targeted tissues into the bloodstream. And that allows a minimally invasive, essentially non-invasive strategy for diagnostics and disease monitoring. And so a pivotal study has been completed where that indication was explored. The data is being analyzed. If that data reaches the predetermined or preset thresholds, then that would be the basis for approval of this device as a diagnostic tool for enriching plasma-based biomarkers from specific brain regions, and more generally, blood-brain barrier opening for this indication. With an FDA-approved device, the path to drug-device combinations becomes much more straightforward. And I think more pharmaceutical companies and biotech companies are going to be excited about partnering with us and other device manufacturers that can achieve, in a similar, again, bioeffect and monitoring strategy and enhance drug delivery and therapeutic efficacy.
You've been recognized with awards for your contributions to the field. Looking ahead, what excites you the most about where Focus Ultrasound is headed in the next 5 to 10 years?
I'm very excited about the possibility that Focus Ultrasound could be used for both enhanced monitoring and diagnosis of disease and treatment of that disease. There are many diseases, brain cancer being one of them, that are not static or stable, that evolve over time. And you need to understand that evolution to be able to treat them effectively. And brain cancer is a great example of this, where we know if we treat these high-grade gliomas that are heterogeneous, very plastic and adaptable biological foes, we know if we treat them with monovalent or monomechanistic therapies that we may kill or treat the cells that are driven by those mechanisms. But the cells that are not repopulate the tumor and grow back. And that's why these tumors recur almost uniformly. So we need ways to monitor this evolution in real time and adapt our therapies with the adapting disease, and potentially allow us to really treat these in such a way that we're hitting them from multiple sides at the same time, not allowing them to evolve in the way that they do against a single therapy and turn what is otherwise a universally fatal brain cancer into a chronic condition that's manageable. That's a very exciting idea in a group of patients that really hasn't had a new therapy for really over 2 decades that's meaningfully changed survival.
Well, thank you so much for talking to us about all the exciting and impactful research you've been doing. We are so excited to keep up with what's next for you and your team.
We are very excited too. And Allison, thank you so much to you and your team for putting this together.
This episode of Curing with Sound has been presented by the Focus Ultrasound Foundation. If you'd like to learn more about Focus Ultrasound or the foundation, visit our website at fussfoundation.org. You can also sign up for our newsletter and follow us on social media. Be sure to subscribe to the Curing with Sound podcast so you don't miss the latest information about Focus Ultrasound. You can also rate and review the show.